Hi, everyone. Welcome to the first episode of the Editors in Conversation for mBio. This is a show about the tiny organisms shaping our world and the scientists that study them. I'm your host, Marvin Whiteley. I'm the editor-in-chief of mBio, and I'm really excited about today and about the first episode because we're going to be diving into a topic that, at least when I started thinking about it and first heard about it, it really sounded like science fiction to me, but it's grounded in real science. And the topic is really what happens when fungi that live in, on, and around us, what happens when they adapt to living in a warmer climate. And I know a lot of you, if you're out there listening right now, you probably think immediately your mind goes to the HBO series The Last of Us, right? Which is this show that really focuses on the zombie ant fungi. So this fungi that infects ants and modulates their behavior, and then these fungi begin to infect humans. And so I'm really excited to have our first guest here. It's Dr. Arturo Casadevall. He's the Bloomberg Distinguished Professor at Johns Hopkins University, and he's actually the founding editor of mBio. And so it's kind of I've been really excited to take over from Arturo since July, but it's been a little bit of a curse too. It's a lot of work to take over a journal. So you have a lot of appreciation for what Arturo did for 15 years. I've known Arturo for a really long time, and he's one of the most creative and fun people. So I'm really excited. We're going to have a really good conversation today. And he's the leading expert on fungal pathogens and spent decades studying how these microbes adapt. And so, Arturo, glad we could get you scheduled, and thanks so much for joining us.
Marvin, so thank you for having me. Glad to be here. Before we get into subject matter, I do want to thank you for all your work as an editor. Editors are essential to the proper working of science, yet it is one of those jobs that is kind of a thankless job. Nobody's happy with you on it, and yet it's essential. So I know it's a lot of work, but I want to reinforce that it's really, really important, and I'm glad you're there.
Yeah, I'm really enjoying it too. So, and I think, just a real quick thing is I flew to Baltimore to chat with Arturo before I started. And, really, I think it's this idea that, like you said, you need someone there that, will really put the time in like you did. And I get a lot out of it too. You learn a lot as you read through the paper. So, well, today I thought that we'd start out with kind of a big picture question, and this is a question that I usually ask to my introductory microbiology class, and that question is just, what are fungi? And usually I get two answers when I ask that question to my students. The first is bread mold, right? It's the very first thing that someone always says. And the second thing is someone says beer, right? Those are the two things that people always say, but it's a lot more than that, right, Arturo?
Well, I think it's a great answer, because in one hand you can see their destructiveness. They wreck your bread. On the other one, is their usefulness. They can give you beer. They can give you wine. They can give you great foodstuffs. Mm-hmm. So it is I think in those answers it encapsulates a lot of the kingdom.
Yeah. And so, how many species of fungi are there now? Have we recognized?
So we know that about 300,000. Okay. But based on genomic analysis and things, that it is estimated that the number is probably somewhere between 4 and 6 million. Okay. And that will make it the largest kingdom in the biosphere.
Yeah. And, I sort of, one of the things that for folks who may not be microbiologists is they're eukaryotes, right? So that's a sort of interesting aspect, very similar to us and very different from bacteria.
Right. In fact, so they have a nucleus. They're so similar to us biochemically that it makes it hard often to find antifungal drugs. Mm-hmm. But the one thing that you got to know when you walk on the forest and you see a mushroom is that they are Animalia's closest relatives. We're a lot closer to them than we are to plants, than we are to bacteria. We're really, really far away from bacteria. But the fungi and the animals are relatively close. Their biochemistry in fundamental is very similar.
Yeah. So one of the things I'd like to start with, Arturo, before we really get into fungi and the warming planet, is tell us a little bit about your journey. You and I have chatted about it a lot. We have kind of similarities in our journeys in science, and what sort of led you to be a scientist, and then and what sort of brought you to fungi.
What led me to be a scientist? I didn't come from a scientific family. I was, as many people can tell by my accent. I wasn't born here. I discovered science in college. I had known, I didn't come from a scientific family. I began taking science courses, really liked it. But somewhere around the second year, I realized that you could do a job doing research. And then, I got the science bug into me, and that's why I've been so interested in.
Mm-hmm. And what about fungi? How did you get into fungi?
That's much later. My interest in science are quantitative. My Ph.D. is in physical chemistry. So I always, my entire life, been interested in making measurements and understanding things at a deep physical level. And then what happened is that I also did medical training. And when the 1980s was a time in which AIDS had just come in, there was nothing you could do except treat the opportunistic infections. And a lot of these people were dying of fungal diseases. So all of a sudden, fungal diseases became really important. And what really got me into them is one experience. I took care of an individual when I was an ID fellow, in which we had cryptococcal meningitis, and I gave them 3 months of amphotericin, and the amphotericin could not clear the infection.
And the amphotericin is an antifungal that's often used.
Right. Also known as amphoterrible in those days, because of all the side effects that it had. But to me, the realization of host failure, what we were seeing here was an individual who was losing the battle because the antifungal drugs was just not enough. And to me, that was the moment in which I decided to start working on the fungi.
Mm-hmm. And I remember, and in the 1980s with the HIV individuals, I remember that this was sort of a real oddity, right? That they were having these infections with these fungi that you just didn't normally see. Maybe cancer people or people with immunocompromised, right?
That's correct, Marvin. I mean, fungal diseases were very rare, and they are very, very rare in immunologically intact humans. Generally, if you find an invasive fungal disease, the first thing people think, well, is there an innate problem of immunity that people didn't realize in this individual? And then all of a sudden, this disease appears out of nowhere, and it begins killing people. Mm-hmm. And the one thing I will tell you, and it gives me tremendous hope in science, is I have seen it. When I began, the life expectancy of anyone with AIDS was less than a year. Mm-hmm. And today is a normal life. Yeah. HIV infection. And that came from basic research, clinical research, investing. And to me, that is such having lived through that is such an optimistic view that tells me that if we keep going today, you can't do anything about Alzheimer's, for example, many of the cancers. But I believe that for everything, there will be a similar trajectory. But we have to keep at it, basic science, research.
Yeah, I use this with my students a lot. We talk about basic science and how it's really important in the backbone for translational research, and I use the HIV case a lot because it's just really amazing to think about those small molecules that have now been developed, right? And and you just have amazing treatments now, right? And it was all from basic research, and while we don't have them, like you said, for some of the other diseases, I mean, HIV was a tough nut to crack too, right? It wasn't an easy one.
If you had told me in 1985 that a day would come when a small molecule would be able to treat a virus that was integrated into the cell, mm-hmm. and that the immunity would come back, I would say, Star Trek. It's science.
Yeah.
This is not going to happen in my lifetime. And yet it did.
Yeah.
And yet it did. And I saw it.
Yeah. Yeah. And that's one of the first cases where I really started thinking about fungi when I read about these fungal infections that AIDS patients were actually getting. And last night I was talking to my wife about the podcast, and Debbie, and I was like, well, we're going to have Arturo on tomorrow. And she was like, and we're going to talk about fungi. And she was like, when is the first time you ever saw a fungi? Do you remember the first time you actually saw a fungi?
Yeah, it would have been a mushroom that I stepped on it, but I thought it was a plant.
Yeah. That's right. I mean, I was thinking about it with myself. I lived on a farm and we had a bunch of chickens, and we had these dry oats that we would feed them, and they didn't like them, but if you'd soak them in water, they would eat them. And so, but and we'd have them in these barrels in Texas with wood over them, and in the summer it's really hot, and they would evaporate below the level, and the sort of the oats were kind of exposed. And I remember opening once, and I opened it, and there was this field of mold, right? And these beautiful, and I remember I was maybe 6 or 7. I just remember thinking, oh, those plants grow fast, right? That's what I kind of thought, So I guess it was very similar to what you're doing. So I guess today, the topic is, the basic idea is that, that the planet's warming and the fungi are going to adapt to these warming temperatures. And so I guess the first question is, what do we know about what kind of impact that can have on human health?
Temperature is going to affect all life on Earth. And when we're talking about fungi, would also apply to bacteria, would also apply to parasites and all of that. Organisms will either have to adapt or die. There is really no way out. And what people often say, well, but the Earth has only warmed about 1.5 degrees. What are you really talking about? I said, no, you have to think about the number of really hot days. When it gets really hot, the temperature in the soils, the temperature on the sidewalk, the temperature in the buildings gets very hot. And this is going to be selection events on it. And I think that we already have data that the fungi are adapting to higher temperature. In fact, last week, our laboratory published from here in Baltimore that if you compare the fungi from the cold neighborhood and the warm neighborhood, the fungi from the warm neighborhood are more adapted. This is the work of Daniel Smith, who had a very creative way of how do you recover fungi from the sidewalk?
Yeah.
He figured out something called this, uses Star-Kist, the candy that is really sticky.
Yeah.
He figured out, it pushes it right on the sidewalk, takes it out and puts it in buffer, and then it dissolves. So what's going to happen to humans?
So hold up. So he's using Star-Kist candy to enrich for fungi?
Yes.
Okay.
Yes, exactly. But you know what? You could use the same thing to pick up bacteria.
Yeah.
Spores, anything on the fungi. And by the way, they are sterile. He did the control. That is, if you get normal Star-Kist and you dissolve it, no, there are no fungi.
Oh, wow.
But I think it's ingenuity. I mean, think for a minute, how would you pick up stuff from the sidewalk? What are you going to go on with a Q-tip?
And so did he focus on sampling on days that were particularly warm, or just kind of just?
He focused on sampling in the summer, but what he did was more creative than that. He went to a cooler neighborhood and a warmer neighborhood.
Mm-hmm.
And a warmer neighborhood, cities are heat islands, as you know, they're 5-6 degrees above centigrade. And what he found is that when he looked at the same species in the warmer neighborhood, they were heat adapted. That is, they were much more resistant to heat shock and to ability to grow. And so within 2 miles in one city, medium-sized city, you already see fungal adaptation. Now multiply that through the entire planet.
Oh, wow.
And all the ecosystems of what's going on. And I think that we are going to be seeing major changes in the microbiology. These are or all microbiology. It's going to happen to bacteria. It's going to happen to small nematodes. It's going to happen to amoeba.
Mm-hmm.
But the net result, I think, is going to be that the organisms are going to get greater temperature tolerance. And here is before we go to another question, and here is the issue. We don't think of our temperature as a defense mechanism.
Mm-hmm.
And the fact is that we being so warm can keep out an enormous number of microbes that simply can't tolerate it. And if microbes then adapt, well, we may see some of them defeat our thermal defenses.
Yeah, I think it's interesting. We don't really think about that as much as we probably should. But I mean, Pasteur was thinking about this over 100 years ago. He had these human pathogens and trying to infect chickens, right? And a lot of them just wouldn't grow in chickens, right? And they couldn't get infections, right? Because the temperatures are different, right?
Chickens are a lot warmer.
Yeah, they're a lot warmer, and these bugs, these bacteria didn't really like the chicken model. And so he wrote an entire paper about that. It was to choose models, you really have to think about temperature and these other things when you think about models. So, yeah, I mean, we've known a lot about it, but I think it's something we haven't focused on a lot. In that,
I don't think that I ever thought of temperature. The big insight came about 15 years ago when we did an analysis of fungal thermal tolerances, and we found that the great majority of the fungal world does fine at ambient temperatures. But above 30 degrees, the number of species that can grow rapidly decline, such that for every degree above 30, you can exclude about 6% of fungal species. That's a tremendous protection that you have just walking around.
Yeah, that's right. Yeah, so I see that, the paper you're alluding to that you just published was some of the work that your lab's doing in urban settings. Is there a reason that you focused on urban settings versus going out into the country and looking at soils? And
well, I think that for one thing, it's convenience. You can walk to your sites to do it. But the other big deal is that urban centers are going to be extreme places because the temperature in our concrete world is just so different. Our cities are heat islands. So if you're going to look for rapid adaptation, I think you want to go to an accessible place that is having an extreme event. Yeah.
And so these fungi that become more thermal tolerant, do they become less tolerant to cold, or?
I don't know. We didn't quite look the other way. That raises a very interesting problem, and that is there are whole sets of fungi that are cold adapted.
Okay.
For example, a fungi in the Arctic, fungi in Antarctica, and the question is, are we going to see their extinction? Because the ability to adapt, if you're cold adapted, it's going to maybe be really hard to climb out of that.
Yeah, I guess I'm just thinking about Baltimore. It gets pretty hot in the summer and cold in the winter, right? So you're going through these cycles, right? And selecting for these, yeah.
And if you try to reproduce in the lab, you got to think about diurnal cycles. The sites, changing temperature from the night to the day, and it's a constantly moving target.
Yeah. And it seems like from the human health perspective, we also have a pretty good example, and that's Candida auris, right?
Right. So Candida auris, about 6-7 years ago, we proposed, Dimitrios Kontoyanis and Vincent, we proposed in mBio that it may have been the first fungus that essentially adapted to higher temperatures. So where does that come from? First of all, it's a thought, okay? It's an idea. It's you can't go back in time and prove it that way. But here is the difficult part for me to explain, Marvin. It emerges simultaneously in 3 continents at the same time. That is, it emerges in South America, Africa, and the Indian subcontinent in the years 2010, 2011, and 2012. And the isolates are not related. It's not like somebody took a plane from it. And since then, we have had 3 more independent emergencies. One in Russia, one in Iran, and last year in I'm blocking it, but it's another place in Asia. And these, again, they're not related. And I ask you, what is going on? I mean, this organism was there all along.
Yeah. Yeah, I mean, I've I'm here in Atlanta, and of course the CDC, they've put out a lot of guidelines, and this has been a bug they're really worried about, right? Candida in the hospitals. And I think one of the reasons is because of the high mortality rates, right? I mean, I think it's 30 to 60%. Is that right?
What happens is it tends to affect people that are debilitated. And then it has a high mortality simply because, on it. But the other problem is this thing is hard to get out for reasons that are not very well understood. It attaches to surfaces and then it's hard to get out. My concern, Marvin, is that it's getting into the community. That is, people are now carrying it around. And when are we going to be able to begin to see, for example, vaginitis caused by Candida auris or skin infections caused by Candida auris in individuals that were not necessarily in the hospital? And I think that it hasn't happened yet, but that is my concern.
Yeah, some of the folks in the hospitals I've been talking to, clinicians and other people, are saying they have a really hard time eradicating it. Like you said, they don't know what's the framework and the procedure, right, to actually get it, because it's really tough when it gets in to get rid of it.
Yeah. Absolutely. I mean, it's a problem, but I think so we have argued that Candida auris is the canary in the coal mine. It's still, there is really no good explanation for these emergencies, and there will be more. And a good hint came from another paper that was published in mBio from my Indian colleagues, who showed, they looked at it in the environment, and they found it in the Andaman Islands. And they found it in two places: a beach that was frequented by people and a marsh that was remote. The organism from the marsh struggled to grow at 37 degrees, whereas the other organisms that are already adapted, they grow very well. In fact, the human isolates don't have any problem growing to 42 degrees.
Oh, wow.
So this ability to adapt has been supercharged.
Is it understood, Arturo, why they what the changes are? What are the adaptations that allow it to grow at higher temperatures?
Not for that organism, but for other organisms that people have looked at. That is, the question is, what Marvin is asking is, what changes do you have to make to the cell or to the genome for it to become thermal tolerant? The fungi can accumulate trehalose, for example, in the short term. They can melanize, they can do some sort of metabolic changes. But when people have sequenced this isolate, what strikes me is that you need very few genetic changes to achieve thermotolerance.
Oh, wow.
Only a few changes, maybe 100, 200 mutations or so to do so. So this is not going to be hard to do.
Yeah. I think to some people that might seem like a lot of changes, but I know at least in the chronic infections we work on in bacteria, over a year period of being infected, you can get, 1,000 mutations, even in a bacterium. Yeah. And so 100's probably that's not that drastic of a change. I think that's interesting. So, when we talk about Candida auris, I was thinking last night, I work on antibiotic resistance, and so for bacteria, right? And you see all kinds of studies that are coming out, and a lot of people talk about this. You see it a lot in the popular press, but seems like public awareness for fungal infections and, antifungal tolerance is just not at the same level. Why do you think that is?
I think that we have a blind spot for the fungal world. I think stop for a minute. You the only time you see it is when they either are in your refrigerator or when they come out of the ground after it rains. The rest of the time, the entire kingdom is invisible.
Yeah.
And I think that yet it is critically important. It's a critically important organism for degrading. The biosphere is totally dependent on the fungal world in many ways. I mean, everything is important. Bacteria important, viruses are important, but it's important to think that these things are interlinked. Yeah. And that for reasons that do escape me, they tend to get a lot less attention, a lot less interest.
Yeah.
You see it in microbiology courses. Mm-hmm. Look at microbial pathogenesis courses. You have the bug parade, right? You have whoever lines them up first. You have the viruses, the bacteria, then you do the parasites, and then you maybe one afternoon for the fungi.
Well, I think it's true. Like when I first started teaching introductory microbiology, I only had, one or two lectures on fungi, right? I mean, because you go through a lot, and I've really expanded that out. A lot of this, because I think you're right, there is a blind spot, but I think for the kids that are, sort of in school now, it's going to be a big part of their lives, right? I mean, this is something that's really happening. But, it's not all doom and gloom, right? I think you alluded to it a second ago. Fungi do a lot of good things for us too, right?
Absolutely. And I think we're just beginning. I mean, let's rattle some of the medicines we get from them. We get the penicillins, we get the statins, we get the cyclophilins that are used in transplantation. It's remarkable that they, as a group of organisms, they spend a lot of their energy in making secondary metabolites. And some of these secondary metabolites are some of our most important drugs. They also make other drugs like LSD.
Yeah.
You can, all kinds of things that can affect mood and hallucinogens and things like that. In fact, we don't understand why that is. We don't understand why they make these kind of compounds.
Yeah.
But when you think about that, we have only looked at a tiny fraction of this number of organisms. I would say that there must be tremendously important compounds medically out there that are yet to be discovered.
Yeah. But also they're really important in like turnover, right? Like, I mean, I know here in Georgia with all the rain, the wet conditions, right? And you cut a tree limb down and it begins to rot. I mean, a lot of that's fungi, right? That are in there breaking
The fungi
down.
are the big degraders, period. Their big job in the biosphere is they degrade and they return elements back into the biosphere that are kind of needed. What they do, though, that when they cause disease, many of these organisms, which are degraders in the environment, use the same approach in the body. For example, Aspergillus ends up putting out a lot of stuff that necroses the lungs around.
Yep.
And then it becomes very hard to treat because dead tissue, you can't get drugs into it. So these things are kind of related. But I think the overall good that we get from the fungal world far, think about bread.
Yeah. Well, beer and wine too, right?
Beer, wine, bread.
So we get all the good food from it too. But if we sort of move back, one of the things I did want to touch on is, why is it hard to develop antifungals, right? Compared to an antibiotic? Like what are the major problems?
I think if you can put it in one sentence, I think it's that you can find more differences between humans and bacteria than you can find between humans and a fungus. Now, you can find them. We don't have cell walls, for example. Well, then we have the echinokindins that are for the cell wall. It's interesting that one small metabolic difference, that is, we use cholesterol to keep our membranes fluid. The fungi use ergosterol.
Oh.
And that small difference biochemically leads to two major classes of antifungals, the polyenes like amphotericin and the azoles like fluconazole, things like that. Mm-hmm. But there are increasingly, people are finding other targets. So there clearly mushrooms are not people. There have to be a lot of other differences. So I think that there are increasingly more antifungal drugs in the pipeline, and it shows you, the human capacity for finding what is needed. But Marvin, you work on antimicrobial resistance, and there is a relationship that needs to be better explored between the ability to acquire temperature, thermotolerance, and drug resistance. Because one way to look at it is that they're both stresses, right? Mm-hmm. When you hit a bacteria with an antimicrobial, you stress it in some way, and if you stress it enough, it can't repair itself.
Right. Yeah, there are these ideas in Staph aureus, a bacterium, right, that when it lives in the nasopharynx at different places in the nasopharynx, the temperature is different, right? And so it actually interacts with the microbiome in very different ways based on what the temperature around it actually is. And so I think temperature, like you said, we don't work on it, I think, or think about it as much as we should, but certainly, The other thing is not all humans are the same temperature, right?
Absolutely. It's a normal distribution. So some people go through life a lot warmer, and some people go through life a lot colder.
Yeah, that's right. Everyone always says 98.6 Fahrenheit, right? 37 Celsius. But I'm like, if you look at the data where that was originally brought from, like you said, there's a big distribution, right?
Absolutely.
Yeah,
absolutely. And the hotter you are, the more you need to eat to maintain, because
that's right.
Most of our energy. But there is evidence that we are getting colder. There is a paper that looked at temperatures over the last century, and our great grandparents were warmer than we are. And
really, I didn't know that. Wow.
Yeah, it was published a couple of years ago. We should probably find it and put it in the. But it's an amazingly study, because you can't say that we don't know how to measure temperature. Temperature is something we can measure back to the 17th century in some way. But probably what happened is that our ancestors lived in a more inflammatory world.
Yeah,
they were, they carried worms. There were much more latent tuberculosis, things like that. A dirtier world raises your temperature. So we're getting warmer, cooler.
Huh. Well, when you think about the fungi, also, I was looking last night. I didn't really know much about this, but I was looking at vaccines, right? Where are we for vaccines for Candida auris?
We have zero vaccines against the fungi. But the good news is that for every major fungal pathogen, people have identified numerous candidates. So it's not hard to make it. Okay. Experimentally, there are a lot of ways to protect against fungi, probably because all you need is to add a little bit more immunity to a system that is already pretty well oiled for fighting fungi. But the problem arises that then it's clinical development. There are few patients, so the economics become more difficult. The clinical trials become harder.
Yeah. But, I guess getting us back to The Last of Us, and thinking about it, so, if we think about what could be a pandemic in the future, what do you think about what should we be doing right now? What kind of surveillance should we be doing? What are the steps that we should take, right, to sort of begin to monitor this and think about whether this is potentially a reality in the future?
So you brought up a pandemic. Before we go that, is a fungal pandemic possible?
Yep.
The answer is yes. Mm-hmm. Look what has happened to the amphibians that are being decimated by a chytrid. Look what has happened to the North American bats that are being killed by a single fungus. Look what is happening to salamanders. So pandemics are feasible. Now, we as humans, and in our own narrow way, we don't think about that happening to us. But because most fungal diseases are not contagious. But there is no reason that you can have a situation in which a new fungus emerges that sporulates in the lung, and when you cough, you cough the spores out.
Sure.
Guess what you get? Communicability in a pandemic. So you ask me, what should we be doing? Well, to me, we know very little about the natural world, and this is the type of research that doesn't get funded. But we need to map the natural world. We need to know what's out there. I would say not only for the fungi, we need to do it for bacteria, we need to do it for viruses, we need to do it. Yeah. This is how we prepare. And the other major problem is that most of what we know comes from only a few species. Saccharomyces cerevisiae is probably not representative of most of the fungal world, especially after it's been domesticated by humans to grow very nicely in fungal growth. So we need to do a lot of the fundamental work to understand a big mapping effort for the biosphere, and that will tell us primarily what's out there. And if you have a sense of that, then you may be able to see where do we need to prepare.
Yeah, I think that, that's true for bacteria too, right? We're constantly, looking, this sort of basic science, E. coli was fundamentally so important for us to understand biochemistry and all the processes that a microbe must do to grow, or a bacterium must do. But now you even change strains of E. coli. They're very different, right? Just strains.
Right.
And
imagine if all the biochemistry that bacterial bio had been done in staph.
Yeah, right.
What would it look like? I mean, I think the fundamental processes are the same. I don't think that nature invented these things once, but it's the variations that make the big difference.
Yeah, that's right. Yeah, I agree. I mean, this has been great, Arturo. And I mean, throughout this podcast, we've talked about studies and science and the need for basic research and how important it is. And I know that when you started mBio, you've written about a lot about this, and also, you've written a lot about scientific integrity and reproducibility. And I just wanted to ask you the question, how do you think mBio moved the needle, like on this subject area?
I think what mBio, the big change that mBio did for the ASM was that prior to mBio, the ASM had basically segmented by journals. There was the Journal of Virology, Journal of Bacteriology. And then there were some journals that they expand some of it, like applied environmental microbiology. But what mBio we try to do is we try to say we're interested in everything, not only in microbiology, but in the allied sciences, immunology, ecology, and all that. And that, I think, created a bigger sense of continuity. Mm-hmm. That's, I think, more what we did. And notice that many of the new journals that the ASM has launched after that, mSphere, Microbiology Spectrum, are general type journals.
Mm-hmm. Yeah, I think that's now a trend too. I think microbiology is the Microbiology Society in the UK is just launching a new journal. That's a very broad journal called Microbiology Horizons, I think, which should be coming out soon. And they're, again, exactly like what you're talking about, not being topic specific, but just trying to find good microbiology.
Right, everything is connected, and I think that's how it became different. How do we try to do things? In the early days, I remember the comment someone said to me, the publishing experience is terrible. I mean, think about it. You find something in your lab you're really excited about. You work with a student, a postdoc. You write it up. You send it out. You get criticism.
Mm-hmm. Yeah, that's right.
If you're lucky enough to get a review.
That's right. Well, I always tell everyone, it's okay to like things, right? It's okay. And I think as scientists, we oftentimes feel like you have to write something negative, right? You have to criticize things in some ways. And I think I've even seen this on grant panels where someone may love a grant, but they talk about the things that they think are maybe wrong, and then the grant doesn't do as well, right? Because didn't mention all of the great things that were associated with it. And I think, I think there needs to be a sea change in the way we think about papers and grants when we get them. It's okay to be excited about it and like it.
That's right. So the one thing that we did in the early years, it actually didn't work as well later as the board become larger, because eventually as the journal grew, you had to bring more people in. But we try to have very clear decisions. Paper is either in or out.
Yep. Yeah.
And, and that was at the time, and people really like that. Where you are, because a lot of times you send your work in, it comes in, you don't know. And that sense of, if I do more work and I get the paper in, and it's demoralizing.
Yeah, I mean, there's nothing worse than being strung along, right? With more experiments and more, right? And it really is, for the early career scientists, it's just tough, right? To know you've put so much time and effort in, it's just really tough. But, and I do want to touch on this because, I mean, you always, for me, Arturo, you're one of the great advocates for early careers, for science in general, but the early career folks. And I just wanted to give you the opportunity to sort of maybe talk about the future of microbiology and what advice might you have for some early career folks that are, maybe grad students and postdocs and new faculty.
This is what I would say. I would say that in 2025, the single most important thing that anyone can be doing is science. And I tell you why. Humanity is about to go through stresses that are existential, whether it is the climate, the failing green revolution, pandemics, rocks from outer space, you name it. Our best insurance policy is science. And therefore, what I would say is careers in science are going to be hard, period. But it's always been this way. But the one common denominator is persistence and determination.
Yep.
And I think so I would say stick to it, keep working. You may not think that your work is related to one of the existential problems. And I would say, no, you don't know that, because all science is connected, and you don't know what contribution you make, where that's going to end up, or how that's going to be picked up by somebody else. And I guess, Marvin, the best example I give to people is, the person who isolated Thermus aquaticus from a hot spring.
Yep.
Who would have known that the simple isolation of Thermus aquaticus, which one day revolutionized criminology, would lead to an ancestry.com, would lead to
that's right,
to a complete revolution by having a thermostable polymerase.
Yeah, I think that's it. And I mean, I think you hit on what I try to tell, folks when they talk to me, it's persistence, right? And, I feel like sometimes, when they see maybe us, they think, it's probably just a smooth go, to be a scientist, but it wasn't. We both had times. I mean, I know there were times I thought, man, I just don't know, that you doubt yourself. Right. And I think being able to, if you have people around you that are supportive, I mean, it's really critical to have people, but you got to be persistent and keep going, I think.
Absolutely. And what I would say is, you and I are pretty established, but my papers get trashed. My grants get unscored. A lot of bad days. You just pick up and you keep going. That's what I would say.
No, I think it's true, right? Everyone always says, "Oh, you never have that." And I'm like, I have that all the time. Like I have these things all the time. Actually,
if anybody can visit here in my office, I have up there the first scores I got from a grant I put in. In those days, there were no unscores, so you could have scores going up to 300, 400, and they are about 300. It reminds me exactly where I came from.
Yeah. My thing I show is I had a very early Journal of Bacteriology paper that we ended up submitting 5 times to finally get it in. It has hundreds of citations now. And one of the reviewers said, this paper will fundamentally harm science if it's published. That's what they said. And I remember getting that back. And, but luckily I had folks around me that really, supported me, and they're like, no, go back in. Right. No, we were going to go back in. If you like it, go back in. And that's
absolutely.
Yeah.
Absolutely. Persistence and determination, and things will work out.
Yep. Well, I have a little bit of an odd question now that we're sort of getting toward the end, Arturo. And this is something I do for my undergrads. I ask them this question. And that's after they've had the class. And so if you could have one dinner guest that's a microbe, who would you invite? Which microbe would you invite?
For dinner?
Yeah, for a dinner.
Oh, my God. It has to be something I won't eat my meal. That will be my first criteria. Something I can't grow in the... I think that and let's say that the microbe could talk and could keep me company during dinner.
Yeah,
I would be most interested in something that can grow. One of those things... extremophiles that it sits there and just watches us from it. That would be my dinner guess. Pretty sure, since he can't grow, he can't ruin my dinner.
Yeah, that's right. Yeah, right? Yes. Yeah. Yeah, that's something that mine would be, one of the Asgard, Archaea, right? Like Loki Archaeota or something, right? Like
actually we're not that far away, right?
Yeah, no, that's right. Yeah. So yeah,
I wouldn't mind taking one that, one of those that reproduces so slowly.
Yeah.
That maybe has watched geology grow around.
Well, Arturo, thanks a lot. This has been a really nice podcast, and and our first guest, and my first time doing a podcast. As a host, anyway, it's my first time, and it was really easy with you.
Well, I can tell you, I've done several of this, and you're a great podcaster. I mean, you kept everything going and the conversation flowing. And if this is your first, I can't imagine where this is going. It's going to be a great success.
All right. Well, I appreciate it, Arturo. And so for everyone, that's all we have for today's podcast. And remember, we're associated with the American Society for Microbiology. And, I'm Marvin Whiteley, and it's so nice to have had Arturo Casadevall here. And if you like the conversation, you can subscribe and share it with your colleagues, and stay tuned. We'll have new episodes that are coming up.